This is lecture 4, part 2. We were talking about the heart and the intrinsic control mechanism of pacemaker, autorhythmic pacemaker of the heart. And now we're going to talk about the external control by innervation from the central nervous system. Now, as I've mentioned before, the heart is made up of cardiac muscle, and cardiac muscle is involuntary, unconsciously controlled. So it's controlled by what's called the autonomic nervous system. So motor output from the autonomic nervous system in the central nervous system leads to the heart and innervates the heart. And there are two divisions. There's the sympathetic division and then the parasympathetic division. So the autonomic nervous system is responding to sensory information that the central nervous system is receiving about blood pressure and blood composition. And the blood is processing that information and then sending it out, sending commands out. It sends commands out through the autonomic nervous system, either the sympathetic or the parasympathetic, to change or regulate the activity of the heart rate. And this is shown here. In fact, the center for the sympathetic center, in other words, the part in the central nervous system that the part of the nervous system that is in charge of putting out sympathetic signals is called the cardioacceleratory center, and these are sympathetic nerve signals that go out. I remind you that the sympathetic chain is a 2-neuron chain. The first one is a preganglionic neuron, then there's a ganglionic neuron in the sympathetic ganglia that sends the postganglionic axons or nerves out down to in this case the heart. Sympathetic stimulation of the heart results in increased activity, increased heart rate. The parasympathetic stimulation of the heart results in inhibition of the heart rate, and parasympathetic signals are sent to the heart along what's called the vagus nerve. So if the heart needs to slow down in its rate, then signals come out. It's actually this area of the brain stem, the medulla, where these centers are. And I showed that to you previously in lab 3, that the processing and signals controlling the heart response to changes in blood pressure come out of the brain stem, particularly the medulla. Full name is medulla oblongata, but a lot of people just say medulla. So that's the parasympathetic signals coming out and going along the vagus nerve, and they're inhibitory, and they go down to the SA node and the AV node, and they have an effect there. The sympathetic pathway is different. It again starts in the medulla. It's coming from the cardioacceleratory. It's a vague name, but cardioacceleratory center. These are sympathetic neurons that send out signals down, and you don't have to understand that complicated pathway, but notice that there are two neurons in both the sympathetic and parasympathetic pathway. In the sympathetic pathway, the ganglion where the first neuron meets the second one that's located in the ganglia, that ganglion is close to the spinal cord. In the case of the parasympathetic, the ganglia are located down here. In this area here, you see there's the axon is making contact with another neuron, and that's much closer to the end target organ. So parasympathetic slows down the heart, and sympathetic stimulation speeds it up, the heart rate. The activity of the stimulation of the via sympathetic pathway or sympathetic division of the autonomic nervous system involves release of the neurotransmitter norepinephrine. Parasympathetic activity involves release of the neurotransmitter acetylcholine. Now, these two mediate their excitatory or increasing stimulatory effect or inhibitory effect through the effect that they have on the membrane depolarization in the pacemaker cells. I remind you that in the pacemaker, what you have is a unstable, constantly depolarizing membrane potential that hits the, let's pretend that this is the threshold. Once it hits the threshold, calcium channels open, calcium rushes in, and then it depolarizes rapidly, and then it repolarizes, and then this process occurs again. Again, you have what's called an action potential, and it comes down, and then again. So these have an effect on that depolarizing, spontaneous depolarizing activity of the pacemaker cells. The pacemaker cells, they're specialized cardiac muscle cells, but they're non-contractile. So they're specialized pacemaker cells. They are cardiac cells, but they're non-contractile cells. They're present in the sinoatrial node. And this is the effect that they have. Normal resting heart rate is shown here. You have when it hits threshold, you get an action potential, depolarizing, then repolarizing, in other words, going back down towards negative, and then starting again, depolarizing, hits the threshold, and another action potential. The effect of, and unfortunately it's not shown here, but the effect of sympathetic stimulation is shown in red, and the effect of parasympathetic is shown in blue. So this is sympathetic activity. If sympathetic activity occurs, you get faster depolarization, and so the heart rate speeds up. And if parasympathetic occurs, then it slows down. And the effect is, in fact, on the rate of membrane depolarization. If you look here, if you have sympathetic stimulation, the slope of the depolarizing action in the SA node, the slope is steeper and reaches threshold faster. And then you get the action potential. The action potential looks exactly the same as the other, but the slope of the spontaneous depolarization is much faster. So the time period in a normal resting heart rate, it's from here to here between action potentials. But with sympathetic stimulation, you get a change, and it's much shorter period between. You see that length is shorter. Similarly, in similar manner, but completely opposite with parasympathetic stimulation, the slope of the spontaneous depolarization of the SA node is much lower, so it takes longer to reach threshold, and then it would come down and again longer to reach threshold. Okay. In this way, the system is regulated. If you have sensory information coming in that the blood pressure is too low, then the heart would have to put out more blood, pump more blood out. In other words, cardiac output has to go up, and so the heart rate goes up. So you get sympathetic stimulation, the slope of the depolarizing change in the pacemaker increases, the slope is greater, is higher, and it hits threshold faster, so the heart speeds up. There are many receptor organs throughout the body, not just baroreceptors in the aortic bodies and carotid bodies. You also have sensory information coming in from other areas. You have chemoreceptors in the aortic bodies and carotid bodies, and that's what they're referring to here under this. So there are chemoreceptors, and they are responsive to PO2 and PCO2, the partial pressure of oxygen in the blood and the partial pressure of CO2 in the blood. You have baroreceptors, as you already know, and they're responsive to blood pressure, and they're sending sensory information into the central nervous system, into the medulla, into the cardiovascular control centers, right, of the medulla. And you also have information coming from, well, the stretch receptors are actually those baroreceptors that we talked about. You know, if the pressure is higher, then it stretches out the elastic blood vessels in the aorta and the carotid arteries, so it causes the stretch receptors there in the aortic bodies and carotid bodies to be triggered. So you have information, sensory information, also coming from those stretch receptors, and which are the baroreceptors, and they're sending information, sensory information, up to the central nervous system, to the medulla again. So you're monitoring both the quality and the pressure. You know, you're monitoring both the PO2, the PCO2, and the blood pressure constantly, and information is going up to the brainstem, the part of the brainstem called the medulla, and that area processes, this area here, processes the information and will send out the appropriate signal, either increasing parasympathetic output if it wants to decrease the heart rate, or it will send out sympathetic signals if it wants to increase the heart rate. And it can do that by sending sympathetic signals directly to the heart, or in addition, it can send signals to the adrenal glands, and the adrenal glands will secrete epinephrine into the bloodstream, and that will go to the heart and have the similar effect in speeding up the heart. So you always have this system of sensory monitoring, processing here in the medulla, up here in the medulla, and then appropriate output to the heart. There are other there is other sensory information. You also have chemoreceptors in the medulla itself that can also respond to the partial pressure of gas, O2 or CO2, And on top of that, the main part of your brain is made up of this area called the cerebrum. Okay, all of this is the cerebrum. And this is where higher control mechanisms are, and more complex, less basic, primitive kind of responses. So high-level processing can occur there, and other commands can come out. With stress, say you would have a higher a triggering of increased activity of sympathetic output. So you always have this monitoring going on. Regulation is by a sympathetic nervous system, which can be stimulated by, from signals coming from the cerebrum, or because of exercise. Exercise will send information into the brain also, saying that the muscles are shortening and lengthening, and the tendons are shortening and lengthening, and the joints are moving. So you should increase your sympathetic output. The body's sensory system is telling the central nervous system, well, your body's moving, you need to increase output from the heart. And this is mediated through the epinephrine and norepinephrine neurotransmitters. The parasympathetic is mediated by release of acetylcholine and has the opposite effect of the sympathetic nervous system. It'll slow down the heart. If the heart is at rest, then parasympathetic tone has a greater effect. So in terms of background resting heart rate, so you have, remember I talked to you about tone, background tone, and the heart is under quite a bit of parasympathetic tone. The autonomic system, autonomic nervous system, is more dominated at rest, at least, by the parasympathetic. That does not mean that that the sympathetic can't override that, but on its own, if you take a heart out of a person, when you do a heart transplant, you are cutting off sympathetic innervation and parasympathetic innervation, and you're leaving only the pacemaker to activate the heart. And the heart beats faster typically because when it's intact in the body, in the original body, and is receiving sympathetic and parasympathetic innervation, it's always getting more parasympathetic when at rest, so you have a higher vagal background tone. It slows down the heart probably to, by 5 or 10 beats per minute. It's not a lot, but it does slow down the heart somewhat. Nevertheless, if you got scared, a car is about to hit you, you'll put out more sympathetic and it'll override any parasympathetic activity. All right, let's talk about the electrocardiograph, or the ECG, sometimes called an EKG. Now, this is not at all the same as the depolarizing and repolarizing activity that you see associated with action potentials that I showed you, that graph I showed you before, taken from the sinoatrial node cells. This is what you see on an ECG is measurement of voltage that can be measured for the whole heart, that can be measured at the surface. So this is affected by, it's not a record of a single cell and the voltage inside a single cell. It is the sum total of all voltage changes that can be detected when the electrodes are at the surface of the body. and is affected not only by the amount of tissue involved, but also by the direction in which the charge is moving. So you should realize that the amount of tissue involved is quite big. After all, with depolarizing wave and action potential in the sinoatrial node, that's, a small amount of tissue sending a signal down to the AV node and then down here. But that is also sent, that action potential also spreads across the entire atria, and that is detected at the surface. So it spreads across from here, across in this direction, across the atria from the right side to the left, and it also spreads down through the atria. And then after a delay of 0.1 seconds, it spreads into the heart, and that's even more tissue. So what you're looking at when you look at the ECG is a record of voltage changes that's a sum total of all of the events that are voltage changes that are happening in the entire heart, and is also affected by the amount of tissue involved and the direction that the charge is moving. Well, this kind of this single tracing here is called a normal sinus rhythm. It is made up of P, Q, R, S, and T. As shown here, you have a P wave, a QRS complex, and a T wave. The P wave represents depolarization across the entire atria. So it also includes the contractile muscle cells that are rapidly, all depolarizing at the same time, functioning all at once because all the cells are connected through gap junctions at the ends of the branches of the cardiac muscle cells. And you get a peak as shown here. You have this let me get an appropriate color. You get this peak here, which is the P wave, and it represents the depolarization across both atria, all the tissue in the atria. And then it comes down, and then you have a Q. And it goes, another peak goes up from Q to R, and then down to S, and that's called the QRS complex. And the QRS complex reflects depolarization of all the ventricle tissue, all the muscle tissue in the ventricles. It's a bigger peak. There's more tissue. Again, it's more complex because it's going in different directions as it comes down and then spreads, from the tip of the heart, the apex and bottom tip, and comes up across the ventricles. Now, after depolarization, you get repolarization, and atrial repolarization is actually occurring underneath this QRS complex, so you can't see it. It's hidden in there, and the QRS probably is diminished in size somewhat because of that. But then you have, following ventricular depolarization, represented by the QRS complex, you have ventricular repolarization. And yes, it goes in the same direction, but again, it's because of the direction of the movement across the heart tissue. Do not confuse this with the pacemaker activity of the SA node cells that's recorded by an intracellular electrode being stuck down into a cell within the SA node, and you have direct measurement of what's happening in that single cell. I should tell you that this PQ interval, whoops, that's no good. I should point out to you that this is, here it's called PQ, but in most places it's called the PR interval. Even though it goes from the beginning of P to Q, it's called the PR interval. And we're going to talk more about the PR interval later. Okay? This whole normal sinus rhythm occurs over a period of about 0.8 seconds. In other words, the heart goes through full depolarization of the atria, repolarization and depolarization of the ventricles, and then repolarization of the ventricles in 0.8 seconds. And if you divide 60 by 0.8, you end up with a normal resting heart rate. So let's move on. That's a little bit about the ECG, very basic introduction. The P wave, as I said, represents not only it represents the depolarizing effect of the SA node, but depolarization of the SA node and depolarization of the atria. I just don't know if this is completely clear here. And atria. And the atria contribute the most to it because they're much more tissue. Then you have a 0.1 second delay before the atria contract. So you get depolarization, then contraction. QRS represents the ventricular depolarization. The T wave represents the ventricular repolarization. And as I said, atrial repolarization is hidden or masked by the large QRS. So in terms of, as shown here across the bottom, in terms of the ECG recording and what's happening in the heart, what you have is with the beginning of the P wave, you get this, that occurs with the initial impulse generated at the SA node. And that quickly spreads across the atria and down to the AV node where it's delayed. And so now that depolarizing effect across the atria is shown here by the whole P wave. Then it gets to Q, and during Q what you have is that you have that delay, and now it's spreading down the bundle of His and the bundle branches and into the Purkinje fibers. So it's now gotten to Q, and with QRS, the QRS complex appearing on the ECG, then that represents the ventricular depolarization and activation, and you get triggered contraction of the ventricles. And then the ventricles relax here with the T wave, which isn't shown, but maybe in the next slide, I don't know. No. So the cardiac cycle overall involves atrial and ventricular contraction, then relaxation, then atrial contraction, followed by ventricular contraction, followed by relaxation. And systole is contraction essentially of the ventricles, and diastole is relaxation of the ventricles. So there are two major phases, contraction during systole and relaxation during diastole. And during diastole, that's when the heart is filling up with. Okay, now in terms of heart sounds, you hear, when you listen to a heart, you hear two sounds. You hear boop, 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 boop. Here they say lub dub. Your book happens to say lub dub. But it's two sounds. The first sound is the sound of the AV valves closing. And the AV valves, which are between the atria and the ventricles, on the right and the left. On the right it's called the tricuspid, on the left is called the bicuspid or mitral. They will shut when the ventricle starts to contract. Now I should tell you at this point, when they shut, all the valves are shut. The semilunar valves are shut and the AV valves are shut. and you get a period of contraction of the ventricles. It contracts around the blood in the two chambers, the right and left ventricle, causing a rise in pressure. And then the pressure rises enough that the semilunar valves, that's the pulmonary semilunar and the aortic semilunar valves, snap open and blood starts to get ejected out. When the blood gets ejected out, there's a back pressure wave that snaps, and the ventricle's essentially empty. There might be a little left, it depends. But then that little pressure wave that, you know, as the blood goes out and hits the blood vessels, there's a little pressure wave coming back, and that helps snap shut the semilunar valves again. And that's closing the semilunar valves. That's the second sound. So the AV valves closing is the beginning of systole, and the semilunar valves closing is the beginning of diastole, or relaxation. If you look at ECG record in terms of electrical potential, you get, you know, this kind of pattern of normal sinus rhythm, PQRST, PQRST. This area here represents systole in here, and this area represents diastole in here. But that also, the main key factor here is looking at the arterial pressure. So if you look at the arterial pressure, it's going to, during diastole, it's going to be low and drop, and then during systole, it's going to suddenly rise up. And remember that the first heart sound is at the beginning of systole, of diastole. At the beginning of systole, and the second sound is the beginning of diastole. So you'll find the first sound occurring when systole occurs. That's the contraction of the ventricles, and therefore the pressure inside, the arterial pressure, is going to be rising, and then you have relaxation, diastole. Whole cardiac cycle about 0.8 seconds long. Most of the ventricular filling is occurring in mid to late diastole, and during that relaxation of the heart, the diastolic phase of the heart of the cardiac cycle, the heart blood pressure is quite low, and the blood is entering the atria and flowing down into the ventricles. Then, and at that point, AV valves are open so that the blood can flow into the atria and down into the ventricles. Then the atrial systole, contraction of the atria, occurs because of the signal initiated at the SA node. Following that, the atria relax, and you start getting a rise in the ventricular BP because of ventricular systole, and that results in closing of the AV valves. And now you get a period where all of the valves are closed, and you have what is called isovolumetric contraction. What that means is it's a closed chamber. All the valves, the two ventricles are closed chambers at this point. All the valves are closed. And so the heart is contracting against a fixed volume of blood, and fluids don't change in their volume when they're under pressure, hardly at all. So iso meaning the same, and volumetric meaning volume, same volume. So there's a contraction around the same volume, pressure rises, the semilunar valves open, and you have ejection of the blood from the ventricles. And then you get a period of time called isovolumetric relaxation. That's early in diastole. Now the ventricles relax, the backflow of blood, that back wave of pressure coming back from the aorta and the pulmonary trunk, snaps the semilunar valves shut. That's the second sound of the bup, bup, bup. There is, in fact, just a funny little notch. It's called the dichrotic notch, but I'll show it to you on the graph. It's a change in pressure because of rebound wave off of the closed semilunar valves. So this diagram has a huge amount of information, and it summarizes everything that we've been talking about. I'll emphasize particular parts of it. So everything here is lined up pretty well so that they're in the right relative positions to each other vertically. You know, the ECG, the heart sounds, the pressure differences in different areas of the heart or the blood vessels coming off the heart, ventricular volume. What you have here is the ECG. Remember that the P wave, during the P wave, you're going to start getting the atrial systole following P wave. P wave represents depolarization of the atria that triggers atrial systole. The key thing to look at is the left ventricular pressure. So the pressure in the left ventricle starts to rise when you get into ventricular systole. So following atrial systole, you get ventricular systole. So it's starting to rise here, the pressure in the left ventricle, and then you get ejection of the blood. The semilunar valves open and blood gets ejected out of the ventricles, and so the pressure is released and it comes down. And then you get that closing of the semilunar valves. So right here is the end of diastole. Now we're looking at ventricular volume. The ventricles are have filled the most. This is volume milliliters. They have filled the most at the end of diastole. So this is end diastolic volume right there. And because you're getting now ejection of blood, the end diastolic volume, the volume is dropping, and you get to end of systole, and the difference between the two is the stroke volume, how much was ejected. So right here, actually, is when blood starts getting ejected. Enough pressure builds up at that point to open the pulmonary semilunar and the aortic semilunar, and the blood starts getting ejected. And so you have a decreasing volume in the ventricles down to the end systolic volume. And EDV minus ESV is equal to SV, or stroke volume. And the different things that are happening in the heart itself are shown here at the bottom. And if you spend a little time looking at these diagrams, you will either go crazy or understand what's happening, essentially. Okay? I would prefer if you understood. That thing, the dichrotic notch, is seen in the pressure in the aorta simply because of a rebound off the closed. Remember, that's closing right here. This is closing of the semilunar valves, and that's increased pressure because the back wave bounces off the closed valves and causes a little rise in pressure. All right. Here is the graph shown separately, showing pressure and volume. On this axis it's volume, and on this axis it's pressure. And you can see that the pressure in the left ventricle actually is in the heart itself in general is very low during diastole. That allows blood to flow into the heart, that low pressure. Flow, any flow of gas or liquid, is always from higher pressure areas to lower pressure areas. If there was not a low pressure inside the heart, then you wouldn't get flow into the heart. So during diastole, the pressure is quite low. There's the pressure in the left ventricle, quite low here during diastole. And then during systole, as the ventricles start to contract, it goes up, up, up. And then here at this point, the green line is volume of blood and should be read off of this axis. That's volume, this is pressure. The volume of blood starts to be decreased because now the pressure has risen up to a level that can open up the semilunar valves and blood is starting to get ejected. And so you go from this point here, which is end diastolic, and blood is being ejected, and you go down to here, which is end systolic volume. And the difference between those two volumes, of course, is EDV minus ESV, and that's equal to stroke volume. That's an E, okay. And then it starts again with another cardiac cycle. They've made it a little bit short here. They've made this cycle only, no, they made it 0.7 seconds. No, I guess, no, 0.8 seconds to here. You know, sometimes it varies. Let's not get into that. Okay, let's talk now about the ECG, some abnormalities. I'll only talk about really, there are many different kinds, very complicated to read, and there are whole books on this to understand ECG. I'm going to explain a certain class of or type of ECG tracing that reveals what are called heart blocks. Okay, just for your information, this top one is tachycardia, refers to a fast heart rate. So here you have, you know, a very fast heart rate, a lot of sympathetic stimulation. It's normal in terms of PQRST. You know, the entrainment of each, the connection between them is steady. Wherever there's a P, there's a QRS and a T. I'm not going to talk about the next one. By the way, just again, for your information, tachycardia is a fast heart rate and bradycardia is a slow heart rate. And, you know, someone who's at rest, who has tachycardia, that's not good. They shouldn't have a fast heart rate. That has to be looked into. But bradycardia is not that uncommon in people, especially highly trained athletes who do aerobic sports like longer distance running or sprinting or swimming, not sprinting, longer distance running or cross-country skiing, things like that, longer distance swimming. They'll often have a slower heart rate. And their heart's so strong, in fact, that it fills and contracts hard enough, it ejects the blood and generates enough blood pressure and blood flow to meet the body needs without beating as many times. You know, some well-trained athletes, and we have students typically every year who have a heart rate, a resting heart rate of like 40. Anyhow, that's bradycardia, slow heart rate, can be quite normal. The regular sinus rhythm you're familiar with, you've already seen that. But sometimes you get a heart block, and I'll go into that not here, but further on. This is the normal sinus rhythm. Every P wave is associated with a QRS complex and a T wave. And every P wave is along this axis, the horizontal axis is time. Every P wave, they're evenly spaced, because that SA node is depolarizing and triggering an action potential through the heart at a steady pace, and there's normal transmission of that through the intrinsic conduction system in the heart. However, and I remind you that if you look at a normal sinus rhythm, it looks like this essentially. You have a P wave, you have a QRS complex, and then you have a T wave, and then you have a P wave and a QRS complex, etc. And I told you that the area from, sorry, that's wrong, that the region from the beginning of P to Q is called the PR interval. On the graph it was marked as PQ, but it's PR interval, mostly called PR interval, as shown here. Now, in a first degree heart block, what you have is a prolonged PR interval. And what that means is that there is a abnormally long delay. Remember that there's a delay at the AV node. There's normally a 0.1 second or 100 millisecond delay at the AV node, so that the signal to depolarize the ventricles doesn't get through immediately, but first the atria contract and then the ventricles contract. So, but it's steady, and there's always a certain, delay. If the delay is longer, then you get what's called what you have is a first degree heart block. So what you have essentially here is that this period here is prolonged from the beginning of P to the start of the QRS, the PR interval in other words. It takes longer. But if you look at the graph here, this ECG tracing, everything is there. You have steady P's, steady depolarizations here, P, 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 P. It may take longer for the QRS to show up, so you do have a first degree AV block, but for every P there is a QRS and a T. So, these kind of people with this first degree heart block, can function quite normally. However, if it gets worse and transmission through the AV node is further delayed so that the PR interval is even longer, then you have a second degree AV block. And what you have here is you have P waves with a long PR interval, relatively longer PR interval, and occasionally there's a P wave with no QRST. Now you look and you see that each of the P waves is steadily appearing at the right time, always appearing correctly, but occasionally, in fact, the signal doesn't even get through the AV node, and so there is no depolarization signal going down into the ventricles, and the ventricles don't depolarize, and you miss that QRST. That's a second degree heart block. And that's more serious, certainly, than the first degree. If that gets worse and the delay is extended further. Sometimes the P wave never gets sometimes the depolarizing signal coming from the SA node never gets through the AV node. And so that's a third degree or complete AV block. And so if you see a tracing like this, I always, and I'll admit there's no T waves here, but that's just because the person or the wave was recorded. But that's not the problem. You have a QRS complex, so definitely there is and the QRS represents the depolarization of the ventricles, and then you have another QRS, so obviously they've repolarized. It's just you don't see the T. Don't worry about that. Forget that. The key thing here is if you look at the P's, you see P, P, P. There's a P under here. It's hidden. P, 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 P. You have steady P waves, so the SA node is depolarizing steadily, as it should. But the entrainment, the in-phase coupling of QRS complexes to the P waves is absent. In fact, the QRS complexes, if you look at them, you can see that they are evenly spaced apart, but completely independent of the P waves. And the reason for that is that just like the SA node, which spontaneously depolarizes till it hits a threshold and then triggers an action potential that spreads down and across, the atria and down through the conduction system, the AV node can also do that. But the AV node does it at a much slower pace. And so the depolarizing effect in a normal person or even first degree heart block, the depolarizing signal coming from the SA node triggers depolarization in the AV node. So it's, its own autorhythmic pacemaker depolarization is overridden. However, if that SA node signal does not get into the AV node at all, you have a third degree AV block or complete AV block, then the AV node will depolarize, will hit threshold, and will generate action potential that spreads down into the ventricles and triggers the QRS depolarization of the ventricles. But it's at a slower pace because the rate of depolarization, the time it takes to hit threshold is much slower for an AV node. And so you have two separate signals, one from in this case, one from the SA node generating P waves steadily, and the other from the AV node generating at a slower pace QRS complexes steadily. But the two are not entrained, the two are not in phase, they're out of phase completely and independent of each other. That's a third degree heart block. Not good, not a good thing to have. And in general, cardiovascular disease is a problem, accounts for about 50% of deaths in North America and Europe. Changes in the blood vessels can occur. Deposits of lipid into the wall and calcium into the wall of blood vessels occurs with aging. It's atherosclerosis. You can also get blood clotting formed on the wall in an intact blood vessel. That's called a thrombus, which could lead to, a heart attack or stroke. These are problems, serious problems. Diet and exercise, the key to good cardiovascular health. I wish I listened to that more carefully. Okay, let's talk about some of the basic physiological functions of the heart in the last few slides. The heart puts out a certain amount of blood, and that's determined, that's called cardiac output, the amount that is pumped out in 1 minute from the ventricles. And it is calculated on the basis of the heart rate times the stroke volume. the amount of blood that's put out. Stroke volume is the amount of blood put out with each beat. However, it can change. It can speed up or it could slow down tremendously, in fact. And you know that your heart can speed up. It actually, at the same time that it speeds up, it typically will also contract harder, and so stroke volume also will go up because there's less blood left in the heart at the end of the end systolic volume decreases. So the contractions actually can be harder. So heart rate times stroke volume is equal to cardiac output, the amount of blood put out in a minute. It can go up tremendously. It can go down too if your heart slows down. And that does happen with sleep, rest, following a meal sometimes. On average, at rest, you have roughly 5 liters cardiac output per minute. As I said, the stroke volume is equal to the volume in the heart at the end of diastole. That's the filling, full volume, and diastole, minus whatever's left in the heart at the end of systole. So EDV minus ESV is equal to stroke volume. Now there's certain factors that can affect stroke volume. One is called preload. When the ventricles fill, they are stretched out somewhat by that contained blood. The more they're stretched out, the more the preload. And the bigger preload will cause the ventricles to contract harder. The contractility of cardiac cells, in fact, contractility is contractile force due to factors other than that filling. So the actual contractility is another factor, how contractile the cardiac cells are. The first key factor in stroke volume is preload, how much the whole thing is filled up with. The actual contractility of the cells, individual cells, how contractile they are. And also afterload is an important variable affecting the stroke volume. And I'll talk about this later, I believe. Well, maybe I should talk about it after. No, I think I talk about it later. If not, I'll get back to it. So preload is the amount of stretching out or expansion of the cardiac muscle before they contract, and it's due to the volume of blood that's coming back into the heart. It's the critical factor that controls the stroke volume. The kind of things that will increase filling of the heart, well, when you talk about filling of the heart during diastole and what the preload level is, you're really talking about how much venous return you have to your heart. And a slower heartbeat and exercise, and often slower heartbeat comes with as an effect of long-term regular exercise, will overall increase the venous return and therefore increase the stroke volume, which is why, with exercise, athletes often have, because they have a bigger stroke volume, they'll have a slower heartbeat. Any loss of blood volume, I remind you, blood volume can change, and it can be due to loss of blood, bleeding. That'll typically, if it's a significant loss, it'll result in a rapid heartbeat. Because remember, cardiac output is equal to heart rate times stroke volume. And if you have lost a lot of blood, then you're going to have less blood returning to the heart, less filling, so end diastolic volume will go down, and therefore the stroke volume will be reduced, and the heart, and because of that reduction in stroke volume with blood loss, because of the reduction in venous return to the heart. There is a drop in stroke volume. Cardiac output will be reduced, and cardiac output, in fact, is cardiac output is in fact a key factor in determining the blood pressure. So you'll get a drop in blood pressure with bleeding. I mean, that's kind of intuitive, I would hope, because the whole system is filled with less fluid. But the main thing you can look at is, in fact, how much venous return there is, and the venous return will drop. Blood pressure, in fact, is equal to cardiac output times peripheral resistance. We'll talk about that soon enough. So cardiac output is a key factor in determining blood pressure, and the blood pressure drops. So with blood loss, you get a drop in blood pressure, and that signal is sent up to the medulla and the brainstem and the central nervous system, and it will trigger a sympathetic response, and the heart rate will rise. So you get a very high heart rate. But if the blood loss is too big, it's not going to be able to compensate enough to give enough cardiac output so that blood pressure rises enough. It's a problem, blood loss. So blood loss is very important in determining, causing an effect on stroke volume. But in the end, that preload is the main, and under normal circumstances, the preload is a key factor. The law, or this feature idea of venous return is called the Frank-Starling law. So with an increase in venous return, you get more filling, you get more stretching, and you get increased actual contractility of the heart. You get a more forceful contraction. Preload and afterload. Blood coming in stretches the heart out, triggering an increase in the contractility. Afterload refers to how much resistance to flow there is. If these are narrowed or they've lost their elasticity in the arterial side of the circulation, then that reduces the ability of the heart to pump blood out. In fact, typically you might even get enlargement of the heart with that, with significant hardening of the atherosclerosis. This is a summary slide showing the kind of effects that we've talked about in this lecture, where we're looking at cardiac output, and the initial stimulus is a drop in blood pressure. I would remind you that regulation of the physiological system their physiological systems are regulated so that when conditions change, if a variable changes, then they can respond to correct it. And here, the change is a drop in blood pressure, and that activates the cardio centers in the medulla of the brainstem. They were called on one slide cardioacceleratory centers, but let's leave that alone for now. Cardiac centers, let's just call cardiac centers. So you get an increase in sympathetic output from the medulla. So sympathetic signals go up, an increase in sympathetic activity. And that will increase the contractile nature or contractility of the cardiac muscle cells. And with increased contractility, you get a decreased end systolic volume. At the end of the contraction, because of the increased contractility of the cardiac muscle cells, there's less blood left in the heart. So if end systolic volume drops, then that means stroke volume goes up. The other thing you have with sympathetic activity is stimulation of the adrenal glands, which will release epinephrine or adrenaline. right? And that increased adrenaline in the blood will increase two things. It will cause an increase in contractility of the muscle, and it will trigger an increase in heart rate. The sympathetic increase in sympathetic activity also directly increases heart rate. It has a direct effect on the heart, so that the heart rate goes up. Remember, the slope of the spontaneous depolarization of the SA node is more steep and hits threshold sooner, so heart rate goes up. I remind you that heart rate and stroke volume are the two key factors determining cardiac output. So in this case, cardiac output goes up. Meanwhile, and we haven't talked about this, but well, I think we'll talk about it separately, actually. Yeah, I don't think we'll talk about this right now. This whole section, I don't think I'm going to talk about right now. We'll come back to that later. So, cardiac output goes up, and blood pressure is equal to cardiac output times peripheral resistance. So, and we'll talk about that later also, but peripheral resistance, and so blood pressure rises because cardiac output rose, and so that corrects this drop in blood pressure, which occurred for whatever reason. Meanwhile, I should tell you that from the cardiac centers, because of that drop in blood pressure, there was, it was sensed, it was incoming sensory information. Not only did the sympathetic activity rise, but parasympathetic activity dropped. When parasympathetic activity drops, the heart rate can also go up in response because you have that background tone, and it's constant, but it's blocked when this happens, this drop in parasympathetic. And so that is the end of lecture 4, part 2.